Welcome to everyone who's listening, and thank you for tuning in. This is the second webinar by MBF Bioscience in the year 2013. My name is Dan Peruzzi, and I'm here this morning with Dr. Susan Tappan. Good morning, Sue.
Hey, Dan. How you doing?
I'm doing well this morning.
So I have recently changed my name. I've given a number of webinars here at MBF Bioscience, but I am no longer Susan Hendricks. So my new name is Susan Tappan. And I'm staff scientist here at MBF, just like Dan, but I also have a role in MBF Labs, which is our contract research organization. And both Dan and I are intensely interested in stereology as a quantification method.
And that's why we're going to talk to you today about stereology concepts and probe selection.
Yep. As always, we love hearing from you both during the webinar and after. So please submit your questions via the question window, and we're going to address them as we go along. This webinar is being recorded and will be available for viewing later.
Okay, so the purpose of today's webinar, this is really not a beginner webinar for stereology. We're not going to go over things like how to follow the rules and how much sampling to do. We do have other webinars about that, and we can help you out with that type of thing. Today's webinar is kind of to take a step back from that, and we want to demystify the process of choosing a stereological probe. We want to give you our advice, tell you our favorite probes, and what probes we think you should be using that are the easiest and the most efficient to use.
Yep, so we're really going to be covering stereology from an experimental design perspective. So prior to sitting down at the microscope and getting going, what do you need to think about, and how is the best way to approach your experimental design? So here's the problem. You want to be able to quantify three-dimensional structures. And that could be anything from the number of marbles in a jar to Christo and Jean-Claude's how much fabric they had to use in order to wrap these beautiful trees in their art installation. So that would be an example of a surface area quantification.
Maybe a volume. Somebody might want to know the volume of those also, yeah.
Mm-hmm.
Here's an odd-shaped figure. It's a star. How could we estimate the volume or maybe the surface area of this solid?
If you're hungry, I mean, it is lunchtime here on the East Coast, so how much spaghetti I have in my dish. And by how much, I mean how long, if I put those strands end to end to end, how long would my spaghetti stretch?
That's one type of thing that can be estimated. Here we have a loaf of marble rye, and it has two components in it, and somebody may want to know what is the volume of one of those components, or what's the surface area of one of those components. So we should try to show you examples from real life of the types of features that can be estimated, but we're going to be working under the microscope.
Right, and so if you take this conceptual understanding of these features that you want to quantify and then apply them to our lives as scientists, we may be interested in quantifying, say, the volume of the cortex, or how many neurons there are in the cerebellum.
Sue and I are both neuroscientists, so we tend to think of the brain a lot, but obviously stereology can be used in any organ. You may be interested in something about the blood supply to the heart, so the length of the capillaries may be of interest. There's a lot of membranes that biologists are interested in. What's the surface area, for instance, the maternal-fetal interface in the placenta?
Right. And so we're going to be discussing all of these in the framework of how do you estimate these 3D parameters on tissue sections. So when you can't observe your entire parameter in its entirety just by looking at it, you need to section it up.
So the solution to the problem, we think, is unbiased stereology.
Absolutely. On the right is one of my favorite cartoonists, XKCD, and this one just makes me giggle when I see it because it illustrates the difference between, say, a normal person and a scientist. So you get shocked pulling the lever, and if you're a scientist, you might say, I wonder if that happens every time. So we're really interested in approaching these types of phenomena as a way to understand it. We want to understand what are the rules that govern how things work. And so in order to approach that, we want to design experiments that allow us to tease apart those those rules or those concepts that lead to the event that we're looking to understand. And so we want to be able to show that a stimulus produces a result over and over and over again, and that is, reproducibility.
And if you use unbiased stereology, there'll be a set of rules that you need to follow, and the rules are not that hard. We're not going to go into all the rules for all the probes, but we do have other sources to help you with that. That will get you an unbiased estimate if you follow these rules so that bias doesn't creep into your sampling. You also need to think about how much to sample, and that's where the precision comes in. And if you can be unbiased by following the rules and do the right amount of sampling to get the precision, like Sue says, we can get reproducibility. The precision and the rules for bias are not going to be the main focus of this talk. What we're going to try to do is let you know how to pick a probe, which one to use.
Right, and stereology is a great quantification method because it makes no assumptions of the size, shape, orientation, or distribution of your feature of interest. And so that really removes that concern that the way that you're looking at your object or your feature is in some way having an impact on the answer that you get.
Okay, so what do we mean by probes? A stereology probe is a shape, a geometrical shape, and a set of rules that guide you in using that shape to, you'll throw the probe down in the tissue, and what we want to do is count events. We want to count the interaction of the probe with the feature that we're trying to quantify.
Yep, so if you were interested in quantifying volume, we'll represent the countable event by the number of points that intersect with your with the volume of interest.
So in that case, the event is the point over the area and the volume, which we show on the left there. Yep. Points for volume. Now, if you're looking for how much area do you have, you want to estimate the surface area, we can use lines. We can count the intersections of the area that you're trying to quantify with the line that you're probing with. The more events or the more intersections of the probing line with the surface, the more surface there is. If we work our way right again and above the picture of the spaghetti, is an example of how we could use planes, segments of planes, to probe for length. So we put the planes down, that's the geometrical shape you're probing for. We count the intersections of the string, that could be a fiber or a blood vessel whose length we're trying to estimate. The more events or intersections you get, the longer the strings are. When you're looking for numbers of particles, what we're going to do is find a unique point on the particle. And so we're going to be using a volume to probe for the number of points that are there. And so this is what we mean by stereology probes, are the shapes that you probe with and the rules that you follow to be unbiased.
Right. So to employ stereology, we've got these probes, but we also need to determine how often we're going to come across the quantification that we're going to be doing. And in order to actually estimate the feature that you're interested in, we rely on systematic random sampling because we do not want to have to visit every aspect of our region or our feature of interest. Instead, what we want to do is we want to do it on a subfraction so that we're efficient. But we need to do it in a way that ensures a lack of bias and provides consistent sampling.
I think another way to say this is we don't want to take just one section through the middle of the region and look only at that. We want to systematically and randomly section all the way through the region, both on the animal level, we see with the sections on the left, and once a section is picked, the XY area should be sampled systematically and randomly. We want to give every section an equal chance of being picked, and every point on that section an equal chance of being sampled. All of the probes we're going to recommend today need to be used with systematic random sampling.
Yep, and there are other sampling methods that are in use with stereologic probes, like the smooth fractionator, for example. So there are other ways to perform your sampling, but they will be used in conjunction with a probe in order to quantify your feature of interest.
So systematic random sampling is important in stereology. That kind of gets at where you're looking, but we also want to talk about how you're looking. So how is that probe, which remember is that geometrical shape, how is that probe going to interact with the feature whose number or length or surface or volume that you're trying to estimate? And we have to think about isotropy. And our definition here of isotropy should ensure that the probe interacts with the feature, giving all positions in the structure the same likelihood of being sampled. We want to be random in three-dimensional space.
Right, and we're going to achieve that in a combination of ways. It could be the way that your object is oriented within the greater structure, so like the cells oriented within the brain region itself. How you prepare the tissue is another key consideration. And then finally, we can also achieve isotropy using the probe.
So the first thing on that list for achieving isotropy is this idea that you can prove that the objects themselves are isotropic. In other words, are the fibers that you're trying to estimate the length of, are they randomly distributed in three dimensions? If the object you're looking for is isotropic, you don't have to worry about making the section isotropic or the probe isotropic. I think that's a hard thing to prove in biology.
Sure. You can imagine an isotropic feature in like a marble. So a marble is perfectly spherical. You can imagine that no matter how that marble is presented.
So that jar of marbles we showed at the beginning, the distribution of those marbles would be isotropic. But take an organ like the brain. If you're talking about the organization of axons, in order to be anisotropic, that axon would have to, on average, come right back to the cell body. And that's not the way the brain is organized. And so most of the features we're looking at in biology, you can't assume that they're isotropic. So instead of assuming or proving that the particle or the feature is isotropic, we're going to either make the tissue isotropic or the probe isotropic.
Right. So you can achieve sampling isotropy in one of three ways. That object orientation, which we say, for the purposes of this talk, is a difficult thing to start off with. So you can design your experiment instead to achieve sampling isotropy either with your tissue preparation or with the probe itself.
Okay, so for the rest of the talk, let's assume that we don't know whether the objects are isotropic, so we have to assure isotropy some other way. One of the ways to do that is by how you section the tissue, and that brings us to this slide.
Yep, and there are three ways that you can prepare your tissue. You can create an isotropic tissue section, and in this case, all three planes are randomized using a 3D spin in the tissue before sectioning. So that would be analogous to that beach ball rolling up there in the upper right.
Okay, and the way this is practically done, one way you can do it is called the eye sector. You actually take your little cube of tissue and embed it in a sphere of embedding material and roll it on the lab bench. And wherever it stops rolling, much like this beach ball is randomly rolling in all three directions, you pick it up and bring it over to your sectioning instrument. And it's a lot of times it's hard to see where your rostral and caudal and dorsal and medial are. So every time you see a beach ball in this presentation, it means that you have had to take the tissue and randomize it in three planes.
Another method is to section your tissue using vertical sectioning. And in this case, two planes are randomized using a 2D spin in the tissue prior to sectioning. So that's why we have the icon of the top.
The top spinning around, randomizing in 2 dimensions. A lot of organs kind of lend themselves to vertical sections because they have a naturally occurring horizontal plane. For instance, skin can be stretched out, and then you can take a piece of the skin and then rotate it randomly. That will randomize 2 planes of the tissue. That's a vertical section.
Right. And then the last option, which is something that is extremely common in neuroscience, is preferential tissue section. So this would be, for example, coronal, sagittal, transverse. In any case where you choose the way that the tissue is presented to the sectioning device, that is called a preferential section. So in order to use stereology, you need to obtain sampling isotropy with one of the other 2 ways, with the object or with the probe. So the reason why you would want to use preferential sections, especially in the brain, it's a very common situation, is because we need to know where we are. And for example, you need to know where the caudate is in order to estimate the number of medium spiny neurons in the CP.
Sure, people have learned to read their tissue in a certain orientation, and many people want preferential tissue sections. For the rest of this talk, if you see a little icon with this atlas picture, it means that you can use preferential sections. If you see a top spinning, it means that you had to go through the trouble of randomizing 2 planes of your tissue section. And if you see the spinning beach ball, it means that you actually had to spin your tissue section, and so it's random in 3 planes. So if you can't prove that your object you're looking for is isotropic, and you don't want to go through the trouble of doing the fancy sectioning like the vertical or the isotropic sectioning, then what do we have to do?
Well, we have to make sure that we achieve isotropy using an isotropic probe. And so if you have an isotropic probe where the probe itself ensures that your feature is interacted with in all possible orientations, then you don't need to prove that your objects are isotropic or make your tissue isotropic.
Okay.
So an example of a feature that would be isotropic would be planes oriented in 3 dimensions, like on the left, or a sphere. Yeah, before we move on, does anybody have any questions about this initial aspect that we've talked about before we start talking about which probes we recommend for each feature?
Okay, we have a question from someone who asks, can these techniques be used for lung stereology? So we are actually going to take each feature, the number, length, surface, and volume, and go through them one by one. Certainly you can use these stereology probes on any organ. I've noticed from reading the lung literature that many times thin sections are used in the lung literature, and so we are going to show you how to do these probes on thin sections. We're also going to show you how to do them on thick sections where you can use the probe itself to be isotropic. So we're going to show you an example of how to estimate surface in the lung on both. It could be used in the lung on both a thin or a thick section.
We also have a question about whether or not you need isotropy in order to do stereology. And the short answer is yes, you need to achieve isotropy in a combination of those 3 ways, either the way that the object is oriented within your organ or structure of interest, the way that you prepare your tissue needs to be achieving isotropy, or the probe itself has to have isotropy. Or you can reach isotropy, that randomness in the 3 dimensions, using a combination of those 3 things that we just mentioned.
And that's a good transition into the second part of the talk, where we're going to take each morphometric parameter and talk about the probes that we recommend, the easiest and most efficient ones, and how isotropy is achieved and whether you can use thick or thin sections.
Okay, so let's get right to it. Let's talk about number estimation first.
Okay, we definitely recommend a probe called the optical fractionator. This probe, when you're estimating number of particles, for instance, there's a picture of 36 cells inside of a volume. It's inside of a stack that's about 40 microns thick. You can see that this is a 3-dimensional volume, and we have particles in there. We need to use thick sections, and by thick I mean 20 to 40 microns thick. We can focus up and down through the Z-plane, and one of the rules of the optical fractionator is to find a unique point on the cell. In this case, we're using the top of the cell. So I'm not going through all the rules for the optical fractionator. We can help you with that. We have another webinar about that. But what I'm pointing out is that in order to focus up and down through many focal planes and find the tops of the cell, here's an example of putting a red dot on the top of the cell in the Z-plane, we need to use thick sections. But notice that if we rotate this block of tissue, and now instead of focusing through the Z plane, we're focusing through the X plane, so that what was the top of the cell is now the side of the cell, which is still indicated by the red dots. We focus up and down and we look for the top of the cells, but from a different orientation. We're focusing up and down through the x-axis. Every particle still only has one top, and that's why the orientation of how you section the material does not matter when you're using the optical fractionator. So if you are estimating, notice how every cell, whether we came at it from the side or the top, each cell has either one red dot or one yellow dot. We've counted 36 cells no matter how we've approached these cells, and that's why the optical fractionator is forgiving regarding how you can section the tissue. You don't have to use the vertical or the isotropic sectioning, and that's why we put this icon of an atlas picture in the lower left, which means you can section the tissue any way you want, the way you're used to looking at it.
Right, and the thick sections actually make it efficient too. Right? But why do we need to compare the appearance of objects through a thick tissue?
Well, the reason you would want to use thick tissue and not thin tissue is so that you can have many optical planes and find the tops of more than one particle as you focus up and down.
So you think the tops are really important, right? And that's because of this.
Yep.
Profile counting is biased, right?
That's right. If you try to do this at lower power and you just looked at the profiles of these particles, the number of profiles would not relate in any easy way to the number of cells. So rather than counting cell pieces, we look for the leading edge, which we can call the top of the cell, and every cell will only have one top, whereas every cell will have more than one cell piece if you're counting pieces of cells.
Right. So from a single image plane, you can't come up with an accurate count of the number of objects.
You cannot. You need at least 2 image planes, and it's more efficient to do this in thick tissue and focus down through those image planes. Mm-hmm. But if you can't, but what if you have thin tissue?
So if you have thin tissue, we recommend a probe called the physical fractionator. And in this case, it's not sufficient that you have thin sections. You also need to have pairs of thin sections. They can be prepared in any orientation because the probe itself provides the isotropy, and the number is the output that you get out of this probe.
So we're still looking for the leading edge of the cells, but we're doing it with 2 physical sections. We look at the first physical section. If the profile is there, but not in the second physical section, we know we found the top of the cell. Every cell has only one top, and we won't overcount cells. But we want to stress that this is way easier to do in a thick section optically than it is to get 2 thin sections, and by thin we mean something like 3 to 5 microns thick, rather than the thick section, which is 20 to 40 microns thick.
Right. So any image analysis program would have to be able to find the top of the object in order to provide an unbiased answer of the number.
And that's easier to do in thick sections optically than it is to do in thin sections physically.
Mm-hmm. Okay.
What if you're working on the bottom of a petri dish, for instance?
In that case, you can use the fractionator technique. So in this way, you would do SRS sampling of a monolayer. This is going to be pretty familiar to people who do cell culture because it's very similar to how you would actually count the number of cells you're going to plate for your plating density using a hemocytometer. In this case, you're going to use the fractionator technique. So your isotropy is provided via the probe, and your output is going to be number.
Okay. So let's summarize estimating number with stereological probes.
So for the optical fractionator, you can have your tissue prepared in any orientation. However, those tissue sections should be thick. The optical fractionator is very efficient because it's much faster to focus up and down at any given sampling site than to do it on more sites on more sections, which is what you need to do for the physical fractionator, where you only have 2 planes that you're making a determination about.
So if you can get thick sections, you can use this efficient optical fractionator.
Mm-hmm. The physical fractionator is similar to the optical fractionator in that you can prepare your tissue in any orientation that you would like. However, you do need thin sections, and they must be paired.
And an example of where you might need this is if you're working at the electron microscopy level, where you can't get thick sections.
Right. So if you're counting synapses, you might prefer to actually use the physical fractionator.
Pairs of thin sections.
Mm-hmm. If you are working in a monolayer, you can use the fractionator method, and in this case, there is no tissue thickness since the cells are associated in a monolayer.
So those are the 3 probes we recommend for estimating numbers of particles.
I want to just take a second and address a couple of the questions that have been coming in. You guys are great. We have a question about lung stereology. There's quite a few people who are interested in lungs, and that's fabulous. So when you have the situation that Ali is mentioning, where they have lung biopsies that are about 2 millimeters to be cut at 2 microns, can we use it for our samples? The answer is absolutely. When you have a biopsy, you have that opportunity to create isotropic sections, very straightforward, because you already have a piece of your tissue. And so in that case, you could achieve isotropy, sampling isotropy pretty easily by just isotropically sectioning your biopsy sample.
Of course, you wouldn't need the isotropy for estimating number, but do keep in mind when you look at a biopsy that you are only finding out about what's going on in the biopsy, and it's not necessarily representative of the whole organ.
Regina asks how thick or thin a section can be, and that's a very important question. When we talk about how thick a section should be, for the optical fractionator, for maximum efficiency and accuracy, we suggest that the tissue section be basically as thick as you can cut it, where you have staining that penetrates the entire section thickness. And so in practical terms, we'd be pretty happy if you could get a 20 micron post-processing section thickness or greater.
And in that case, you could use the optical fractionator, and of course you can use preferential sections.
Yep, and then if we want to talk about thin sections, this idea of thin sections needs to be, it's relational to the object that you're trying to estimate. And so for thin, you want to make sure that the object that you're trying to estimate has a chance of appearing in more than one section. And so for synapses, you would need, sub-micron range.
At the EM level, right? Mm-hmm. And that's one of the reasons why the physical fractionator is so difficult, because we're trying to get 2 physical sections that are closer together than the diameter of one particle.
We'll spend a little bit more time on the physical fractionator, and then we'll carry on. But there's what we mean by paired sections are sequential sections. So if you have your tissue sectioned at 2 microns and you want to count cell bodies, for example, that would be appropriate. And so you could take 2 sections that are sequential and then have an interval between the next pair of sections. And so when you're counting, you're actually counting what appears on section 1 versus what is no longer there on section 2 for each pair of sections through your systematic random series.
Yeah, and the reason you need pairs of contiguous sections is because we need to look in one section and see that the cell is there, and the next section and see that it's not there. Then we'll know that we found the cell top.
Right, and that's actually, it's when we talk about stereology, we want to get you back to being efficient. And so the physical fractioner has specific applications where it is the most efficient method and accurate method for estimating number, for example. But we really recommend that you prepare your tissue where the tissue is cut thick, the staining penetrates all the way through, so that you can be more efficient and get your answer faster.
If at all possible, get thick sections and use the optical fractionator rather than the more difficult physical fractionator.
Yep, and in this case, just one last point, those thick sections, when we talk about 20 microns, it's post-processing section thickness. So after you've actually stained the tissue and it's on your slide, we want a nice thick section.
And you can get a lot of shrinkage, especially with ascending alcohol concentration histology.
Yep, and so that has an impact on how you actually do your implement the optical fractionator. We have a webinar on that. I highly encourage you to visit our website and check that out if you'd like to learn more about the practical application of that method.
So these summary slides are kind of the take-home message for this webinar, and for number, here is our probes that we recommend.
Yep. So let's move on to volume estimation.
Okay, so rather than trying to estimate the number of particles, now we're trying to estimate the volume of a region. And what you can do is use a method called cavallari with point counting. And your job, and again, we're marking an event. In the case of number, it was the top of the cell. Now we are marking a point if we see that point over our region of interest. So we have 4 plus signs on the right. And if I see the point is the vertex of the plus sign, if I see that point over my region, I put a green dot on it. Each point has an area associated with it. And so if I mark 4 points, I am telling the program that there are 4 regions of this type of area on my...
So you're representing each area by a single point.
Yes.
So you're representing an area by a position of a point, and so it's a very straightforward way to get a quick estimate because you are making fewer decisions about the location of
right.
the
So you
region.
have this 3-dimensional solid, and what you do is you systematically and randomly section all the way through it. And now here's an example of one of those sections.
Yeah, and so you can see here that the grid is randomly tossed onto the region of interest, and you would mark, as I did with these round circles, red circles, the points in which the grid points fall over my region of interest, which in this case is the right cortex.
It simply adds together the area associated with all those points and then multiplies by the distance between the sections to come up with an estimate of the volume. Mm-hmm. You can do this on thick or thin sections, so it's very forgiving that way.
Mm-hmm. And in any orientation. Because the isotropy is achieved via the probe, you can prepare your tissue any way you like. And so the estimate that I would get from a series of sections cut in the horizontal plane, measuring the right cortex, versus a series of sections that were cut in the coronal plane, measuring the right cortex, they would obtain a similar unbiased estimate.
In other words, it doesn't matter which direction we section up this region, we are going to get an equally unbiased estimate in either direction, so you don't have to worry about sectioning your tissue in any fancy way when you are using cavallari point counting.
Mm-hmm. If you're interested in the component volume, so for example, plaque load in a neuroscience application, you don't necessarily want to have to sample at this high a density in order to calculate how much of the cortex is occupied by A-beta plaques. Instead, what we can use is a technique that drives off of the cavallari point counting principle called the area fraction fractionator. So like the cavallari point counting, we're going to use SRS sections that can be cut either thick or thin because we're going to be operating on a single plane, actually.
We're putting these points down on a thin image, so we don't need thick sections to do this.
Yep, and they can be in any orientation. So the isotropy, again, is achieved via the probe, and your output is going to be a subvolume. So the amount of cortex occupied by plaques is a way of representing the data that you will obtain using this type of probe.
Okay, let's summarize volume and cross-sectional area estimates.
So for cavallari point counting, you can prepare your tissue and for sectioning in any orientation that you would like.
Great, so we don't have to worry about vertical sectioning or isotropic sectioning.
Not at all.
We can use coronal or sagittal or the way we want to section it.
And it can be any section thickness as well. It can be a single image plane, or it could be a thick or a thin section.
Okay, and the same goes for getting an estimate of the percentage of one type of tissue per another type of tissue, a volume to volume ratio with the area fraction fractionator. Absolutely. So it's very forgiving when we're estimating volume.
It's very simple. You don't have to have a lot of forethought in your experimental design for stereology prior to actually sitting down at the scope to calculate the estimates for volume or area.
Okay, and when we estimated number, we could also go at it from any orientation, but we did have to worry about getting a thick section so we could use the optical fractionator.
Yep, and just a reminder, it's not one representative section that we're actually estimating for the area or volume. Instead, what we're doing is we're having an entire region of interest available for sampling, and then we're going to look and quantify the area on a systematic random sample of those sections through your region of interest.
Okay, always use systematic random sampling. Okay, so now we've looked at number and volume. Let's turn to length and surface. Thanks for reminding me, Sue. Before we go to length and surface, let's look at the volume of particles. So we were just looking at regional volumes, and you may ask, well, why don't we just thoroughly section through the cell or the particle and do point counting on it? And that brings up one of the tenets of stereology, which is do more, less well. So we don't want to find out as much as we can about one cell. What we want to do is look at a group of cells and compare it to another group of cells. And so we're going to use a probe that, rather than sectioning exhaustively through the cell and doing point counting, we can throw down a line and see how the line interacts with the particle, with the cell. So we have this same group of 36 cells, and rather than finding their tops, I want to look at one of them, and I'm going to put a line down. I'm going to pick a random point in the middle of the cell, and then put a line segment down to the edge of the cell, and that's the red line there. So with this nucleator probe, we are actually taking a cross-section of the particle, putting a line down, then we take the length of that line and put it in the formula 4/3 pi length of the line cubed. So we get an estimate of the volume. Because we're working off a cross-section, if we approach this cell from another direction, we will get a different looking cross-section and a different length line. When we look at all the cells in our groups, we don't want all the cells in one group to be cut along their long axis and all the cells in another group to, for instance, be cut along their short axis. So that's why we need to use a vertical section or an isotropic section when we do a local probe. We need to use thick sections because we want to pick these cells without bias to their size or orientation. So we're going to use a dissector, and a dissector, as we showed you when we were estimating number, needs to be put down in a thick section so you can focus through and decide which cells you're going to do the nucleator on. Then when you do the nucleator, it matters how the cells were cut. Therefore, you need to do this thing with the beach ball where your block of tissue is random in 3 planes, or do a vertical section where it is random in 2 planes. And if you use vertical sections, you're going to have to show the software what the vertical direction is, because it's going to sine weight the line and make up for the fact that one of the directions is not random.
So what you're saying is that a combination of the probe and tissue, or actually if the object you are estimating you know to be isotropic, we have to achieve sampling isotropy in some combination of ways for the nucleator. The nucleator itself is not an isotropic probe.
Okay, so you're right about that. If you can somehow prove that the particles themselves are isotropic in space, and that's been done before, we can give you a reference in at least one region where someone has taken the trouble to do that, then we don't have to worry about being isotropic. But we don't want to cut these particles down their long axis every time or down their short axis. Therefore, we need to take the piece of tissue and make it isotropic for the isotropic nucleator, or we need to just spin that tissue to make 2 planes isotropic, and we'll make up for the other plane with the probe, and that's called the vertical nucleator. This is a rule that gets skipped or ignored a lot in stereology. You can always get something from your data. But this is probably the least forgiving of the probes because we need to use isotropic sections or vertical sections, and we need to use thick sections.
And all of this is relevant because it depends on the population of the objects that you're trying to estimate. So if you don't know if they are isotropic, then you have to take pains to ensure that you achieve sampling isotropy with the probe and with the tissue preparation. As Dan mentioned, there are some reports in the literature of proving isotropy of a population of interest. There's a paper by Christoph Schmitz and Patrick Hoff that looked carefully at a population of cells in the striatum, I believe, in order to demonstrate that those cells themselves have no preferential orientation in the striatum, and therefore you can prepare your tissue with a preferential orientation for that region.
But if you don't know about your objects and you don't know that they're isotropic, then we need to use isotropic sections or vertical sections, and we need to use thick sections when we're probing for local volume.
Okay, so we've received a bunch of questions about this next topic we're going to cover, which is length estimation. Length estimation is of great interest for those people who are interested in blood vessels in particular, as well as fiber innervation. So if you want to understand whether or not a tumor has successfully recruited blood vessels to feed it, you can estimate the length of the capillaries to give you a metric for the invasiveness of that tumor, for example.
Yeah, so length is important to a lot of biologists, and this is a tree, a length tree. It's kind of shows the historical progression of probes from the bottom to the top. So on the bottom of this tree are probes that are used. For instance, if you have isotropic tissue, and we're going to highlight, so this is what we mean by this can be confusing, and we're going to try to demystify it. We're going to show you the 3 probes on this tree that we think that you can use for most situations.
Right. So again, these 3 probes that we're going to highlight we think are the easiest to implement and the most efficient and accurate way to get to your answer.
Okay, so if you don't want to prove that your object is isotropic, or you don't want to have to cut the tissue so that it comes out isotropic, like isotropic or vertically, the best thing to do is make the probe itself isotropic. So what we have here is it's something like a 60 micron thick stack of fibers. So we can see that this is a 3-dimensional stack, and we are going to put a surface down into these fibers to estimate the length of the fibers.
Right, an isotropic plane known as a sphere.
Yes, we're going to use a surface, and we're going to the surface is going to be the surface of a sphere, because the sphere itself is isotropic. This probe is called Spaceballs. We need thick tissue so we can get the sphere in the tissue. Like I said, this is about 40 or 60 microns thick. You focus up and down, and wherever you see the fibers pierce the surface of the sphere, you mark that event. So I think we can see right here that a fiber is going through the sphere, and we put a pink dot on it. So here's one space ball, and every place, I think we're going to zoom in on this a little, we can see the fiber going through the surface of the sphere. The more intersections there are, the more the length is. There's a formula that will estimate that for you.
Right, and the blood vessel or fiber itself can pierce the sphere multiple times, and so long as it penetrates that plane of the sphere, you would continue to mark
And
it.
that makes sense. The longer it is, the more it's going to pierce. Don't forget that we're using systematic random sampling, so I've done 6 spaceballs here, but only one of them is showing. The more intersections you get, the longer your fibers are. So we're using thick sections so that we can use a probe that is itself isotropic, and that means we can use preferentially oriented tissue.
Yep.
What if you don't have thick sections? What if you have thin sections?
If you have thin sections, your options are a little bit more limited in that you need to achieve isotropy not with the probe, so you need to get it some other way.
Yes.
And so the most common way is with isotropic sections. So you prepare your biopsy or your sample of interest using the isector method. So you'd create it with an isotropic sectioning technique, and then you can use the image plane itself as the probe.
So we're using a thin plane, and we're counting the intersections of the fibers with that plane that's shown on the bottom. Just like with the sphere, the more intersections you get, the longer the fibers are. But we need to make sure that this thin plane will interact with these fibers in the same way. And therefore, we have to take the tissue itself and make it isotropic, since the probe itself is not isotropic.
And we don't know if the fibers themselves are isotropic. And in most cases, I think more so than cells, I think it would be safe to assume that most fibers do have a preferred orientation, even if they were blood vessels in a tumor.
Especially in the brain. A lot of times you're not going to know that, so use the thick tissue and use the sphere. The probe itself is isotropic. If you do use thin tissue, you have to make the tissue isotropic. That's why we have the spinning beach ball at the bottom.
So if you have a monolayer, the method that you can use to estimate length is a technique called petrometrics. In this case, you do get an isotropy via the probe, and the output is length. And so in this case, you would have like a cycloid shape here that is presented across your tissue as in a systematic and random way, and you would indicate by placing a marker on each point where the fiber and crosses the probe itself.
So usually we use a plane to probe for the length of a line, but because we're in a two-dimensional situation here, we can use one line to probe for the length of another line. I'm not sure if you guys can see these white lines inside of the counting frame, but there are these cycloid lines, and you mark where the lines intersect the fibers. The more intersections you get, the longer the fibers are. Mm-hmm. Should we summarize length estimation? Remember that complicated tree? We want to boil that down to just these 3 probes.
Yep. So Spaceballs is is an isotropic probe that is used in thick tissue, and that tissue can be prepared or sectioned in any orientation that you prefer. If you have thin tissue, then you're going to be restricted to processing that tissue in an isotropic manner in order to be able to use an image plane as your probe. And then finally, if you have a monolayer, our recommended option would be to use petrometrics.
Like if you're at the bottom of a petri dish, that's why it's called petrometrics. Okay, so let's turn to surface area, which is going to have similar concerns to length, in that we're going to need to make sure that the probe interacts with what you're probing for in an isotropic or a random way. And we're going to narrow down this kind of historical tree of all the probes that exist in our software to 2 probes that we like that we're going to tell you about.
Right. There are many options, but we think that these 2 are used widely in the literature and are able to give you your answer in a efficient and accurate manner.
So just like with length, we're going to start off with a thick section so that we can make the probe itself isotropic. This is maybe a 40 micron thick image stack of some choroid plexus. Can we start?
Oh, sure. Sorry about that.
If we start the movie here, I'm just going to tilt it a little so that you can see that this is a 3-dimensional image stack. And we're going to try to estimate the surface of this choroid plexus. Of course, we'd use systematic random sampling. This might be just one of those sampling spots. We can focus up and down through the Z-plane. We're going to use a line to probe for a surface.
But not just one line, right?
We're not going to use just one line. We're going to use a triplet of lines. These lines are going to be mutually orthogonal to each other. So the first line, like the yellow line, is thrown down in a random location with a random angle. And then the red and the magenta line are 90 degrees offset. See how these 3 lines, triplets, are mutually orthogonal to each other. So now we have an isotropic probe. Our job is to focus up and down and mark the event. The event is the intersection of the line with the surface. So we're focusing up and down on that yellow line, and we can see that the surface of the choroid plexus hits the line right there. So we put in an event or a mark on there. The more intersections of the lines with the surface, the greater your surface area is. Notice that we're using lines that are all oriented in 3 directions. We need to do that in thick tissue so we can get those lines oriented mutually orthogonal to each other in that section. But the probe itself is isotropic, so you don't have to worry about proving that the object is or doing fancy vertical or isotropic sectioning.
So in the case of surface area estimation on thin sections, so for you guys out there in the lung stereology world, this is probably a probe that you're a little bit more familiar with. You can use a probe called cycloids for SV. This uses thin SRS sections and gives you surface area as your output. And we're going to achieve isotropy both via the sectioning method and via the probe. So we're going to use vertical sections, and we're going to indicate the vertical axis of the section plane.
That's that yellow arrow, right, Sue?
Yeah, it's the yellow arrow there. So you indicate that, and then the probe itself are those red lines that you see, and you just mark the intersections of the probe with your object of interest, which in this case would be the membrane of the choroid plexus.
Okay, so we don't have a thick section, so we can't stick the triplet of lines in there and make the probe isotropic. So we have a thin section. So we're using a combination of processing the tissue, vertical section. We spin that tissue block and we will randomize the X and the Y plane. And then we use a special kind of line called a cycloid, which is going to randomize the third plane for us. We could do a whole webinar just about vertical sections and cycloids, and we can definitely talk to you about that. But the point here is, if you're using a thin section for surface area, you can use a combination of vertical sections and the probe to ensure an isotropic interaction of the line that you're probing with and the surface whose area you're trying to estimate. So to summarize surface estimation, I think, if at all possible, try to get thick sections, make the probe itself isotropic. You won't have to prove that the object is, and you won't have to use a vertical or a isotropic section.
If you are using thin tissue, then it should be prepared with a vertical sectioning technique, and in that case you can use cycloids for SV.
Okay. We just got a question going back to length, and it says, can you clarify why Spaceballs is a probe for length but not density? So we are probing for length, and we're using a surface, and the basic idea is that the more intersections you get of the fibers or the blood vessels with the surface, the longer they are. There's two methods that you can use, and we have not got into this with this talk at all. It's not under the scope of this talk, but you can use a fractionator method, or you can find a numerical density. If you use the fractionator method, you keep track of the fraction of the volume, and then we extrapolate to get the answer. And in that case, Spaceballs can be used that way, and will give you only a length.
And that's a positive thing, actually.
Yeah, I think it is.
If you have concomitant changes in the volume of your region and the length of your structure that you're estimating, if you're using a density measure, you may not be able to ascribe the difference that you see, because you could have a change of both length and volume that would end up giving a density that's the same between your experimental and control.
Yeah,
group.
So what you're saying, Sue, is that we're using model-based stereology. We're taking pains to section all the way through the region, and it's very powerful to say length per region rather than length per volume.
If you do want a density, you can then estimate the volume using Cavalieri point counting methods, and then simply divide your unbiased estimate of the total length by the unbiased estimate of your total volume to get a length per volume value.
That's why it estimates length.
Yep. And Cavalieri, as I hope we've illustrated a little bit, is Cavalieri is very straightforward to do. It's a very simple. It doesn't take any time really to actually estimate the volume of your region, and so recommend using a probe like Spaceballs or the optical fractionator to get your number or length without calculating a density.
If you actually do need a density, so think about that question too.
Yep. One last thing about length is that tissue shrinkage. So the way that you prepare your tissue is of extreme importance for length estimation.
The estimates we'll be getting will be on the tissue as the artifact has left it.
Yep. So you want to minimize your shrinkage as much as possible, because we know that the Z shrinkage is greater than XY shrinkage when you're preparing tissue sections. So bear that in mind, and be prepared to present your data as a post-processing estimate.
Okay, so we've gone through this whole talk, and we haven't really mentioned the word bias too much. But I hope that you realize that the things that we've been doing, the concepts we've been describing are to avoid bias. And one of the ways you can get into bias is where you look. And so we really wanted to stress systematic random sampling and the idea that one histological section is only going to tell you about that one histological section and not the whole region.
Right. You want it to be representative of the whole region itself, not the way that you're looking, but the answer that you're getting. And so you need to have the whole region available for sampling in order to ensure that any changes that may be happening in different locations are observed.
So all the probes we've recommended, you're going to want to use systematic random sampling, and that's a big advantage too, because you can get a good snapshot, a good estimate of what's going on by counting surprisingly few events.
Mm-hmm. And so that's how you achieve some efficiency for your quantification. This is, and then you obtain this lack of bias by achieving isotropy.
Yes, this whole concept of isotropy that we talked about. Look through the webinar and look at those summary slides. Those are going to be your take-home messages for when you're trying to estimate number or length or surface or volume. That's going to let you know how you're going to assure isotropy and whether you can use a thick or a thin section.
Yep. Every aspect of your feature has an equal opportunity of being visited by your probe, and that's really what we mean by isotropy. All positions in your structure have the same likelihood of being sampled. We're going to say that sampling ensures that your estimation is representative and precise, and that we're going to encourage you to choose a probe that's efficient and easy to implement in order to obtain that unbiased quantitative information that you're looking for.
And that was the criteria we used for these probes that we recommended. And as we went through this talk, I hope that this idea behind stereology to do more less well also came out.
Right, but do more or less well does not mean an incorrect answer. So let's just hark back to what you gain by thinking about the answer you want during your experimental design phase and choosing stereology over a method like ImageJ or something like that, where you're going to use an image analysis program to count cell profiles. Cell profiles, because it doesn't represent a unique point of your cell, means that you are not ensuring that you're going to count an object once and only once. Not only that, but that cell profile is going to have a bias depending upon the size of the object that you're looking for.
Okay, and keep in mind that if we don't get to your question today, we're going to go through them and answer them, and please contact us to talk about today's topic, which is how do you decide which probe to use, or more specific topics like how do we follow the rules for this probe and how much sampling do we do.
Yep, we have numerous webinars on our website already, and we're going to continue doing these, especially if you let us know that you like them, and feel free to offer up topics that you'd like us to address. The practical demonstration webinars cover how to implement the optical fractionator and Spaceballs, as well as Cavalieri and area fraction fractionator. You can refer to the primary literature in order to see how researchers in your field are using stereology to obtain unbiased quantitative estimates of their feature of interest. And we also have how the methods were developed as references within our software, Stereo Investigator. If you'd like to learn more about Stereo Investigator or contact us with questions, you can contact us for a free trial, and you can also just give us some feedback. Let us know what you'd like to learn more about. Thanks for your attention.
Thank you very much.